If Jonathan and Richard and Petra can join us up here. So while you're coming up, there's sort of this dichotomy in the pediatric population between some of the stuff, Jonathan, where is Jonathan? He's there, that you said about, we should never refer to these first-in-human trials as therapy. Kathy, you too, yes, of course. As therapy, yet, to go into a pediatric population, there's a requirement that there is a chance of therapy, and how do you deal with that when you're thinking about consent forms, when you're talking to families, when you're talking to foundations? That does seem like sort of impossible to fulfill both, and I assume you don't think that no trial should be done in children, but how do you deal with that, Jonathan, first, and then our other panelists?
Sure. So thanks. So this may be a heterodox view, but then again, 90% of my views are heterodox. I actually think the regulations that the U.S. has put researchers in a real bind. I think they really kind of force researchers to shoehorn a lot of really, really important research into a therapeutic category where I'm just not confident it really belongs in that therapeutic category. There is this other category that exists, the 407 mechanism, so studies that can't fulfill the previous the other sort of three risk categories. For various reasons, that mechanism is rarely invoked. I would argue that perhaps it ought to be invoked more frequently. I would say that there are exceptions to my general rule that we ought not to consider participation in early phase or first-in-human trials therapeutic. They'd be exceptions where we have exceptionally good mechanistic knowledge, so that would be perhaps in some of these conditions in gene therapy. Also conditions where, again, if you think about that intervention ensemble, where we have really, really good, strong reason to think that we are not on the other side of the window of a subtherapeutic dose. So we're quite confident. That would typically be something where we already have experience with similar kinds of interventions, and we know the dose range and the toxicity of those similar interventions. So, in ethics, there are no sort of hard and fast rules. It's about sort of thinking through the particulars, but I do think there are some sort of exceptional cases where it could be plausible to make a case where these would fulfill 405 or 5052, is it? The therapeutic justification FDA regs.
Sure, Richard, do you want to talk about dealing with this issue with your patients and trial design?
I think in the clinic we find it when there is no treatment for a disease, and we have the prospect of a gene therapy, which is where retreatment is challenging at best. And these parents are desperate for anything. They'll sign up for anything. So the real obligation, I think, is on the physician who's serving as the investigator here to frame the discussion very carefully and to look at the risk and benefit. So at my institution, we've instituted a policy where we have a patient advocate that comes in, and actually when we're dealing with children and infants, which is what I do, that they really try to make sure that I'm presenting things in an unbiased way, that the parents are absorbing this, that they're not going to sign on the dotted line for anything without really understanding what they're getting into. And that has served, I think, quite well to sort of frame the discussion and sort of slow it down and make sure that they really understand the risk, the potential benefit, particularly if they're coming in at that first dose, which is the one that is creating the most, I think, concern about is there really any potential benefit here? Hopefully, yes, we've selected a dose that, as you heard from Kathy, will provide some benefit, if not optimal, but something. But that, I think, is the big challenge that parents really don't particularly appreciate, I think, that side of it. They'll sign up for anything.
This is one of the most challenging questions, and I think in SMA we've seen that certainly we fully understand the urgency, but then as a result, sometimes parents were led to have with when we did repurposed drugs, for example, with valproate, they were just trying. Of course, to do the best for their children, but it turned out that actually was not only not beneficial, but it actually cause challenges to mobility because it caused weight gain and children actually lost motor function. So I think it's very important to, be really very transparent and have really good information to the parents to help them understand that sometimes just trying something in an uncontrolled way or getting to the sort of N of one ultimately can slow things down and can also have safety issues. So I think it's a really challenging conversation to have, but I think the more we partner with patients, the more we have really, which increasingly happens, really strong patient groups who can provide that kind of information to parents and patients, the better off we are. I think we have to see that we're all in a way, partners in this, and our goal is to get these much needed treatments to as many patients as possible, understanding the urgency, but at the same time doing it in a way that we're actually getting answers and getting data that we can interpret and are not left with more uncertainty and more safety concerns potentially.
So I don't want to take up all the questions. Anyone from the audience like to bring up some topics for discussion? Hello?
Yeah,
hi. Introduce yourself and say where you're from.
Krish Roy from Georgia Tech. I'm also a member of the forum. I just wanted to broadly ask you, I'm an engineer, not a clinician or a biologist. So when I see a lot of the clinical trials data, I feel that there is always inadequate patient data that's collected in clinical trials, especially large clinical trials, that lets us assess or stratify who will benefit and who will not benefit in the future. So for example, we know very little about the immune status of the patients or detailed disease progression models and so on. So we can take the therapeutic characterizations and map them into patient characterizations and eventually learn from clinical trials as to what the stratification should be. Does that something that go into considerations when you guys are designing trials? And in many cases, it's difficult to collect a lot of data from patients, especially pediatric patients. But where does that balance go between collecting a lot of patient-specific data, because all patients are going to respond differently even if the product is an AAV vector of some kind. So how do we reconcile that? I don't know. Anyone?
I think that's an excellent point. I think the more information we can collect, the better we understand. This is early, I think, still early days for some of these therapies, and there's a lot we need to, I think, very carefully monitor to see which patients would be most likely to respond. I think there's excellent efforts, for example, with some of the critical path initiatives where, data from completed clinical trials are being de-identified and are being, provided potentially to other researchers.
But also to your point, I think, is we study fairly narrow slices of the broad population. And how generalizable are the results, even when they're highly effective? Well, what about the older child or someone with a different phenotype? And so it's an iterative process, clearly.
And does that go into a post-market data collection initiative? Especially for the companies.
Yeah, clearly, there's a requirement for long-term follow-up, and many gene therapy trials have these kinds of registries, very important. Again, where the partnership with families and patients comes in, because obviously, what a gift to go for 15 years of your time, but also so important for the field and for others. So that's very critical.
Right, and then also to broaden things into the real world. Once the drug is commercially available and you can get real-world data, then you're not looking at the efficacy in the study, but the effectiveness in more of a post-marketing phase 4 type study. So the importance of having registries that are going to collect patients going forth into the real world, I think, is particularly important.
Dwight Copel, Duke University. How can we fully discuss the issue of the patient only having one opportunity for gene therapy in the current situation, especially in the early phase, low-dose situation?
We have a session that's going to be focused on this exact issue, I think, as well later, but we can talk about it some now, but we That's kind of the theme for the day from some of the later speakers.
So your question is, how do we address the issue of that you may only be able to receive the therapy once? I think the only way to do that is through the informed consent process. And I think that I believe that actually a lot of the people who are presenting for inclusion in gene therapy trials now have a fairly sophisticated understanding. So I think the question revolves around, do you want to be part of the dose escalation study, or do you want to hold back and wait until the investigators have identified the dose that they think is best to take forward? But, fortunately, there are people who are willing to enroll even when there's less certainty about going forward. I think both of the trials you heard about here today actually did a pretty good job of identifying the first dose to be administered based on animal studies, and they were able to identify a dose that actually did convey some biological benefit, but that won't always be the case, I'm sure.
Hi, Chris Saha, University of Wisconsin-Madison. Thanks for some very exciting presentations. I wanted to ask a pretty broad question about new types of tools that you see in this space assisting with this question of getting to minimum effective dose. It sounds like animal models, as well as, in some cases, historical natural history databases and initiatives. What if you were to try to identify 1 to 2 types of tools or models that could be disseminated such that the stories of SMA or RP65 could reach other disorders and diseases, what would those be?
Well, I just have to say that I'm personally very fond of large animal models because I think that they have been really useful in predicting therapeutic doses. I realize that not every disease has a large animal model, but I think Brian Casper and his colleagues did a very nice job of developing a pig model of SMA through a knockdown. So I think my experience has been that it's difficult to extrapolate from mouse studies to determine an initial dose.
I think in addition to efficacy, that probably also, I would say is the same for safety. To also have large animal models is very helpful that are as close as possible to humans. And then in parallel to the non-clinical work, to work early enough with the patient groups, with the investigators, to make sure that there is trial readiness and not just things that when we were in academia, they were published, but things that actually go beyond that and are actually really useful for clinical trial planning. That would be really helpful, working with the patients, hearing what matters to them, and working with the investigators prospectively.
Yeah, and I would add just a small comment that we're finding the greater utility in developing informative biomarkers for early readouts, both for safety and toxicity and efficacy. So we're putting more emphasis on that as well, even in the situations where we have these drugs and they're working. But as you know, there are responders that work well and less responders, and we're trying to sort all that out. So I think that'll be important going forward.
I think you were
Michael Ferenzi, GSK. A number of you mentioned that there's a need for frequent assessment and visits. I was wondering what sorts of technologies or sensors have you looked at for sort of continuous monitoring, like watches or other kinds of sensors that would continuously monitor the patients?
Certainly a really important area of development that we need to explore, especially when we get to very rare diseases where patients may have to travel for the treatment, but then, in the long term, especially, for many years, it would be wonderful if we could have measures that could be done remotely, certainly, or even that would be more naturalistic in the patient's environment. And we're looking at that right now for some of the indications in our pipeline, where we're working with digital measures, and there's many of them. There's, of course, all the measures of like accelerometers that can measure activity and gait. There's measures that can look at voice. There's things that can look at manual dexterity that can all be done with digital devices. So I think that's really the future. The issues there are often the data interpretation. How do you deal with the flood of data? And also patient privacy. So we would ideally like, in some cases, something that's in the patient's home. And although many people now have all kinds of devices that scare me personally, where you tell some device to turn the light on or off, but when it comes to research, there's of course a whole different level of scrutiny, and that's really kind of in the way right now, if you wish. But it certainly is something we have to work on as a field, and I don't think this is a primary outcome in the near future, but certainly something we are exploring, and a lot of issues to be overcome, like the flood of data and the privacy concerns.
And I think also when you're dealing with rare diseases, the patients are coming from a large distance often to the study site. So we're exploring in our study group trying to do more things through a telemedicine approach. Mm-hmm. So they still have the same number of visits, but perhaps every third visit is actually on site, and we do others more remotely.
So, and I would just add that for neurodegenerative diseases and for adult neurodegenerative diseases like Huntington's or even things like DMD, to try to assess that from a clinic visit where somebody comes from a long distance and they have to do some performance test versus collecting data from wearable devices over a longer period of time in different settings and so forth. But somebody is going to have to correlate the data that they develop from wearable devices and so forth with more standard measures. And I think that's going to be one of the challenges.
So sometimes gene therapy...
Identify yourself.
Sorry, Rosemary Hunziker. Sometimes gene therapy is more like our traditional way of looking at drug development. And sometimes it's a cell therapy, which has an altered genetics, and then the cell gets put back in, which is a little bit more like personalized medicine because it's often derived from the patient and then put back. So I was wondering, from a procedural and ethical point of view, have you found any, or have you thought about or identified differences in the way we have to approach these things when it comes to trying to get them approved and accepted? Are there differences in the way you have to look at these things based on whether they're more like the traditional drug pathway or biologics pathway, or the sort of like new ideas in cell-based deliveries?
Well, I think we could probably spend a whole day devoted to that question, but I think that a very important, you're making a very important point, which is that many of these therapies, many of these cell therapies, require a very substantial infrastructure. What you heard us talk about was AAV. AAV is a lot like other specialty pharmaceuticals. You manufacture it, it gets shipped to the pharmacy, and they thaw it and suspend it in whatever is the right thing, a syringe that goes to the OR or an IV bag for SMA. Cell therapies require a much more complex infrastructure. They are typically administered in the context of something that looks like a bone marrow transplant. And one of the things that I think about sometimes is that if one of these cell therapies for sickle cell works, okay, there's 70 to 100,000 people with sickle cell disease in the United States. And in the U.S., we typically perform about 25,000 bone marrow transplants a year. So what are we going to do for infrastructure if somebody develops something that works? So I think it's a very important question, and I don't have the answer.
And I think that there are quite a few gene therapy using cell therapy products coming in the next session, so some of that can be discussed. But I also think the issue of the ability to do more than one treatment and not completely ablating your chance for the next great thing, has the advantage maybe with cell therapies, if you think about it that way, that you could collect cells and store them. They're often stored as backups, but potentially people need to think about for some of the newer, less well-studied approaches such as gene editing, that maybe the autologous backup needs to be, possibly releasable for another try with a different treatment if the therapy fails. Anyway, Jennifer, I think we have time for probably just the people standing up now.
So
before coffee, which we all need.
I'm Jennifer Puck from UCSF. And this may be a little bit related to the last question, but we've heard about a selection of very rare diseases and also mention of an N equals 1, very, very rare disease. And of course, we know that there are hundreds or thousands of disorders that could be potentially treated with gene therapies. And rather than start each one from zero, could there be classes of gene therapies that could build on the established leaders, the first ones? For example, all retinal diseases could maybe take off with a springboard from the starting ones, or certainly bone marrow therapies and other therapies. So we don't all maybe have to have a large animal model if we have a similar other disease. And I want your comments on it.
Well, I'll start by saying that if I did not believe that clinical development timelines would shorten as the field progresses, I don't think I could come to work every day. But I do think that we've seen that over the now nearly 30 years of clinical trials in gene therapy, that as more experience is gained in a particular avenue of investigation, the timelines for development do shorten. And the development of all those guidance documents that were issued last year and more this year has certainly, I think, helped to make things easier for sponsors. And I think that we can expect more and more of that as the field progresses. In terms of ultra-rare diseases, I do think it's important to remember and recognize that not everything has to be taken to the point of product approval. There are things that are treated for a long period of time under perpetual INDs. And if it's an ultra-rare disease, you don't necessarily have to go all the way to product approval.
Kate Lorenzen, University of Connecticut, and a member of the Forum on Regenerative Medicine. So first, hello, everyone. And I guess my question is, let me just start by saying I think that gene therapy has really entered this renaissance period, and it's really great to see it happening. And also, to quote Yogi Berra, I know it's tough to make predictions, especially about the future. But my question is, where do you see it in terms of gene therapy going in terms of 5, 10, 15 years from now, in terms of the non-rare diseases? I mean, there are people who are looking at osteoarthritis in my area and looking at gene therapy types of methods. And you're probably one of the more optimistic groups that will be here today. So tell me what your thoughts are about where it is, not in terms of rare diseases, but where are we in terms of gene therapy being a mainstream for treatment of non-rare diseases in terms of, for example, osteoarthritis or other diseases?
It'd be good to have a crystal ball, but I think that at this earlier stage, and this will change over time, we have to look at the risk-benefit ratio. And I think at this time, given the uncertainty, it is probably in the near future more likely that gene therapy will focus on, in a platform approach in a way, on areas where there is increasing understanding of the vector and an increasing understanding of the safety profile. And initially it'll be severe diseases that are clearly monogenic. And then hopefully over time, as we understand the opportunities of the technology better, as we get more confidence with the safety, we could think about other diseases that are more chronic diseases. So one thing is the risk-benefit profile. The other thing is the scalability, and that was alluded to already. So I think at this point there's also a lot of innovation needed in manufacturing processes and so on that would make it challenging right now. So I think it'll be a process over time, like with so many new technologies. And you could think about some of these conditions where you could use vectorized antibodies or approaches that are quite different from the gene replacement approach that we've seen right now.
Can I just add one thing to that? I mean, it is one of my most cherished hopes that we will see the extension of gene therapy past single gene disorders to genetic strategies for complex acquired disorders like osteoarthritis, like congestive heart failure, like age-related macular degeneration, and I could go on and on.
Like cancer, we're already there.
Cancer, we're already there. But I do think it's going to be important for us to be able to integrate, for example, large data sets around genotyping so that you select for the trial the patients with that complex acquired disorder who are most likely to benefit from your genetic strategy for this disease. But I definitely think that we will see that, I would say, within the next decade. Who's the trial start?
Great, thanks.
Okay, last question, Nadia?
Yes, Nadia Lomelsky, NIH, NIDCR. I want to ask about this particular diseases discussed today. In terms of the stability of the therapeutic response, like Dr. Hayes showed 2 years, do you know anything about future? Can you predict from the mechanism of action what to expect? Because if it will be 2-3 years, it's good, but what about 20 years? And maybe 20 years you re-administer, it won't be as much immune response. I mean, how do you form your strategy on the basis of this consideration?
So one of the slides that I skipped for want of time was on long-term follow-up, and I think that for gene therapy trials, there are really two reasons to do that. One is to look for delayed adverse events, but of course the other is to get a sense of the durability of the effect. And so for the clinical development program for Luxturna, for the patients involved in the phase 3 trial, we have committed to 15 years of follow-up, and very generously, the patients have been coming in and doing the 1 or 2 days of testing that are required to get all of the endpoints. And I think at this point, we have data for everybody up to 4 years and some people through 5 years from the program, and we've been submitting all this data every year to ophthalmology conferences and showing the durability.
But can you tell something from mechanism of action, what should happen?
Well, the cells that the vector went into, the retinal pigment epithelial cells, are long-lived cells. So it will almost certainly depend on the lifespan of those cells in people with the disease. And I think one of the challenges for early studies in gene therapy is that we typically populate our trials with all comers, but you can imagine that the time will come when almost all the patients who receive the therapy are young, newly diagnosed patients. And I assume that the younger you get people with genetic diseases, the better your results are going to be. And so, I think we have to remember that too.
We do have a session this afternoon on long-term follow-up study issues and who funds it and how do we convince patients to do it, et cetera. So I think it's time for coffee. Thanks to everybody.